Hi, good day everyone. So today we are going to discuss the SCL named Deadly Dust from BSM4301. Our team consists of me, Faiq, Sarah Lisa, Hani Sophia, Edina RAUQAH, and ZIKRY. So first and foremost, we will begin our discussion by explaining each of the group's study case. Now, let's begin with the first study case.
Okay, so now I will be explaining about the first case was to identify the pathogen from an unknown biological sample. The sample contained three unknown proteins called protein A, protein B, and protein C. And them we use plasmid, which is a of millions of known protein sequence in a database, which it also gives an E value, where a value close to A is race. This protein bacteria control important processes such as gene activity and protein function in different environments. Protein B was identified as Calderia cynospecie. Its main the vector to infect the body and form biofilms that protect them from the immune system. Defined as the else protein helps the vector obtain Calderia cynospecie, both and tarsis. The protein C were more close combine this result with the patient's history of being exposed to a suspicious powder. Um, this was the most likely pathological and track. So to summarize this case, it shows how plastic can identify iron proteins and support disease diagnosis by combining laboratory result with clinical information. That is it for case one. Thank you.
Okay, thank you. Let's hear from case 2.
Oh, okay. My name is Sarah Lisa, and I have been asked to do the case 2 report. So, where we needed to use a fibroblast to identify the unknown protein and determine the pathogen responsible for the infections. So, here's the background. Mr. Rodney, a lab staff member, died after opening an envelope containing a suspicious white powder. Before he died, he showed symptoms resembling a Klebsiella phenomena infection and left a streak clue, which is a conserved pattern and accession number of OON70417.1 and the word maybe. For this task, we need to use a fibroblast because we had both a sequence and a conserved motif to search with. And a fibroblast is specifically built to find sequences containing a defined motif pattern. The motif pattern we used was K followed by K, followed by G, followed by E, followed by L, followed by I, followed by H, followed by one of A or S, and followed by A, followed by P. For our method, we took the query sequence from the accession number and defined this conserved motif pattern and submitted both the fibroblasts and analyzed the significant matches from the alignment. Our results showed four set domain containing protein, all protein lysine and methyltransferases from Bacillus cereus, Bacillus parenthesis, Bacillus falciparum, and Bacillus sp.ssr7, and all with identical and highly significant scores with 100% identity and 100% query coverage. So our top hit was Bacillus cereus with a score of 150, 100% identity and 100% coverage. For statistical, the e-value was 8 times 10 to the negative 44, which is far below the 0.05 significant threshold, meaning that this is a highly significant match. And in terms of clinical relevance, Bacillus cereus is a non-opportunistic pathogen associated with food poisoning and wound infections. Even though the symptom resemble a Klebsiella phenomena, that organism was excluded since no significant alignment was detected. So the clinical picture was misleading. For our group conclusion, fibroblast analysis point to the Bacillus cereus group with Bacillus cereus as the closest match, making it most likely pathogen. And this also fits the white powder clue since Bacillus species form a spore. So that's all for case report 2.
Thank you. Let's move on to case report 3.
Hi, my name is IRDINA. So right now I'll be presenting about the third case study. So this case began with the death of Mr. Rodney after he was exposed to a suspicious white powder. He experienced symptoms such as shortness of breath, vomiting, and a high fever, raising suspicious that the powder contained a harmful biological agent. Therefore, our objective was to identify protein X using the SciBlast tool and determine whether it was a toxin or simply a protein domain, as well as identify the possible pathogens. So first, we analyze the protein sequence, which consists of 131 amino acids. The sequence contains both hydrophobic and hydrophilic amino acids, suggesting that it forms a folded globular protein. Since it is relatively small, it was suitable for comparison against the NCBI protein database using the SciBlast itself. So the SciBlast analysis shows that the protein X has 99 to 100% sequence identity with proteins from Bacillus cereus group, including Bacillus cereus and Bacillus parenthesis. With 100% query coverage, the analysis reached its final result after 4 iterations where the top match was identified as a set domain containing protein from Bacillus cereus ATCC14579. In addition, the e-value of 2 times 10 to the power of negative 27, which indicates that the match is highly significant and not due to random chance. Now, based on these findings itself, we concluded that protein S is not bacterial toxin, but rather a conserved set domain containing protein, which is believed to function as a protein lysine methyltransferase involved in protein methylation and the regulation of cellular process. In conclusion, the SciBlast analysis successfully identified protein X as a set domain containing protein with a very high sequence similarity to protein from the Bacillus cereus group. The result including 100% query coverage, 99 to 100% sequence identity, and a highly significant value of 2 times 10 to the power of negative 27, as I stated before, provided a strong evidence that protein X is a conserved protein involved in cellular regulation rather than a bacterial toxin. So that's all from my case study.
Thank you. Let's move on to case 4.
Okay. For my part, which is case 4, I will be presenting. So where our objective was to identify an unknown pathogen using T plus N analysis and phylogenetic analysis because we got the protein X sequence. So first, let's look at the case background. The patient developed chills, shortness of breath, nausea, and vomiting after opening a suspicious letter. The condition rapidly progressed to severe lung inflammation and respiratory failure, eventually resulting in death. So this clinical history strongly suggests exposure to a dangerous airborne pathogen. Then I will explain about the phylogenetic analysis. The phylogenetic tree compares the unknown DNA sequence with several bacillus species as shown in that tree. They have six bacillus species, which is Specifus, Parenthesis, BINTI, Thuringiensis, and Tracis and Series. So the as shown in the tree, the unknown sequence cluster most closely related to Bacillus anthracis, although it belongs to the Bacillus series group, which contains closely related species. The close clustering indicates that Bacillus anthracis share the highest genetic similarity with the unknown sample. Based on both the molecular analysis and the clinical symptoms, we conclude that the most likely causative pathogen is Bacillus anthracis, the bacterium that causes anthrax. Specifically, the patient's severe respiratory symptoms following the exposure to a suspicious letter that are consistent with inhalational anthrax, which is the most severe form of the disease and has a high mortality rate if not treated promptly. So for my conclusion, I think the cause is um, Bacillus anthracis. So that's all from our case for.
Thank
Shopee.
you. Now move on to the final case, which is case 5. Hi, my name is Faiq. I'll present about case number 5. So for case number 5, the background is as follows. We have Mr. Rodney, which opened a suspicious envelope containing a white powder. After that, he developed symptoms after 2 days. The symptoms are high fever, shortness of breath, and persistent vomiting. And he also mentioned that those symptoms are quite similar with infection due to Klebsiella pneumoniae. And after several days, he is found to be passed away. And after his death, he left several clues. The first clue is a sequence pattern, which is KKGELIH, AOS, and AP. And another note containing a series of containing an extension number of OON 70417.1. And we are instructed to use fibroblasts to analyze the clues given and to find out the pathogen that is responsible for the cause of his death. Doing fibroblasts, we search for the extraction number and we copy the FASTA result to the BLASTP. Using BLASTP, we choose the database name and guidance quality sequence and we choose the fibroblast algorithm. After blasting the program, we found several sequences that have significant alignment with our clues. The top four is Bacillus cereus, Bacillus brandtricis, Bacillus specificus, and Bacillus anthracis. Among those four, we found out that anthrax, caused by the main protagonist Bacillus anthracis, is responsible for the murder or for the death of Mr. Rodney in the form of anthrax spores. Upon inhalation of the anthrax spores, it will present a severe pneumonia, pneumonia-like respiratory distress, such as high fever. And we also found that Bacillus cereus causes can cause gastrointestinal illness that lead to vomiting symptoms. So, for our group's conclusion, we conclude that the fibroblasts managed to identify the unknown protein as a sex domain containing protein. The closest sequence match were Bacillus cereus, Bacillus brandtricis, Bacillus specificus, and Bacillus anthracis. And the sequence alignment indicated that the isolate belongs to the Bacillus cereus group. And to finally conclude it, based on the sequence analysis and clinical symptoms and forensic evidence, Bacillus anthracis was identified as the most likely causative pathogen. That's all from case 5. Now, since we all have presented our case, we must decide on the pathogen that is involved in our case study. Anyone wants to continue this discussion? From what I have noted, the study case 1 showed that Bacillus anthracis was the culprit, while study case 2 and 3 concluded that Bacillus cereus was the pathogen that is the main culprit, while case study 4 and case study 5 says that the main pathogen is Bacillus anthracis, which and all of those two type of pathogen is in the group of Bacillus cereus. But nevertheless, we need to choose which pathogen is the one that is responsible for it. What do you guys think?
Hmm. Aduh.
Because two of the group says Bacillus cereus, while the other three says Bacillus anthracis. So,
So we have to choose two to decide either of these two, right?
yeah, we have to decide between the two. Which one is it?
Mm.
I think because majority of us suspect the Bacillus anthracis, I think we should choose Bacillus anthracis as the suspected pathogen. Do you guys have any other suggestion or opinion?
I agree with Hanie's opinion. Everyone okay with that? Okay,
Uh,
Yes, I agree with that.
Yeah.
then we can conclude that the culprit in our study cases is Bacillus anthracis.
yes.